everybody, and hello, Charles. Thank you so much for joining me today as we go down the rabbit hole and take a look at some of the weird and wonderful ways that genomics is being applied in research. Today we are going to be looking at how genomics is being applied in relation to psychedelics and research in that area. So Charles, if you could just introduce yourself and tell us a little bit about what you do as well.
Hi, thank you for speaking to me today, Shannon. I'm Dr. Charles Nichols. I'm a professor of pharmacology at LSU Health Sciences Center in New Orleans in the USA. And I have been studying psychedelics professionally for almost 25 years now, predominantly as serotonin 5-HT2A receptor function in psychiatric diseases, and more recently as anti-inflammatory targets.
Yeah. So my knowledge in this area is zero, essentially. So I think it'd be great to kind of start off with some a brief summary of kind of what psychedelics are and kind of what their effects are and kind of the pathways that they work on. If you could kind of give us an overview of that.
Well, psychedelics are a class of drug that activate what's called the serotonin 2A receptor in the brain and throughout the rest of the body. But when they activate this target receptor in the brain, it produces this distortions, sensory distortions, dissociations from reality, the psychedelic trip. There's magic mushrooms, there's LSD, or there's some of the more infamous psychedelics that are out there. And they're very, very potent and powerful drugs that, due to, I think, political considerations in the 1960s, 1970s, were really made illegal to research through the scheduling drug laws. And so not really much work had been done on them from the 1970s, 1980s into the 1990s. There were only maybe a handful of people that were doing preclinical work. But now they've really come back as we're starting to understand some of the potential medicinal roles for psychedelics to treat disorders like depression, drug abuse, and inflammatory related disorders.
Yeah. So what are some of the kind of social and political issues that you kind of mentioned, and how has that kind of attitude shifted? Like, what has made that attitude kind of change in more recent years?
I think it was a lot of it was in the politics of the 1960s, sort of as a way to control the hippies and the upstarts that were protesting against the government. Is like, well, we can't arrest them for protesting, but they're all using these drugs, so let's make these drugs illegal and then we can arrest them. And they were seen as dangerous drugs with no real medical value, and really a halt to just about any research, both the clinical psychology studies as well as cell culture in vitro animal studies, that it became very difficult to get permission from the DEA and the government to do research on these. So because of that, maybe only 5 or 6 people in the world were really able to do any kind of research on these drugs as initially sort of how are they involved in drug abuse, and to show that. But then we began to understand that serotonin and serotonin 2A receptors are really involved in several aspects of mood and cognition and normal brain function. And then in the 1990s, some clinical studies were performed. There was a study by Rick Strassman using DMT, just characterizing what effects the DMT had on people. And he wrote a book about this, DMT, the spirit molecule. Charlie Grubb at UCLA did the first clinical study of using psilocybin to treat end-stage cancer patients and saw some really amazing therapeutic effects there. Then that was followed on by some studies from Johns Hopkins, Roland Griffith's lab, showing that a single administration of psilocybin had really long-lasting positive effects on affect. And then they followed that up with studies looking at end-of-life associated depression and anxiety at Johns Hopkins and NYU, and then Imperial College in the UK with Robin Carhart Harris and David Nutt. So really prestigious research groups then showed that a single treatment with psilocybin had these profound antidepressant-like effects in these resistant patients. And then popular culture books, for example, from Michael Pollan, came out around the same time. And people now, I think it wasn't so much of a stigma. Back in the 80s, it was don't use drugs, it's going to fry your brains. Well, now we understand that that once we put some effort into researching them, understand that there's some really significant medical potential associated with this class of drug and activation of the target serotonin 2A receptor.
What are kind of the most well-studied psychedelics in the kind of this area, and why are they the kind of ones that are kind of more well-studied than compared to the other ones?
Well, there are several that are found in nature. The most widely known one in nature is psilocybin, which is in the magic mushrooms. There's also DMT, which is found in ayahuasca brew. It's one of the main components there. There's 5-methoxy-DMT, which is in the toad secretions. So they've been used for thousands of years by indigenous populations. Mescaline is in the peyote cactus. But I think the one that was most well known was LSD. This was the one that is derived from ergotamine, which is made by a rye fungus. And it was used by a lot of the counterculture in the 1950s and 60s. So most of the research was done with LSD early on. Psilocybin has become more prevalent recently, I think because of the demonstrated safety of it for hundreds, if not thousands of years in indigenous populations. The psilocybin itself comes from a natural source with very little toxicity associated with it. It's less of a dirty drug. Psilocybin is only targeting a subset of serotonin receptors, whereas LSD lasts much longer, is having effects on other receptors. So I think the focus recently has been on psilocybin in the clinical therapy setting because of its relative selectiveness for serotonin receptors, and its behavioral effects really only last a few hours, 3 to 4 hours, as opposed to say 8 to 12 hours. Although both LSD and psilocybin are being used in the clinical setting, I think the focus really has been on psilocybin.
Yeah. Why is kind of studying psychedelics so kind of relevant, particularly now as we're kind of trying to look for like different types of drugs? Like, why is kind of researching it so important?
There really, depression is really been a significant burden to society for decades, especially now with COVID. The rates of depression, psychiatric disorders have really gone up recently. And there have been no new antidepressants on the market since essentially the 1980s. The last major breakthrough in the pharmacological treatment of depression was essentially Prozac and modulation of serotonin levels in the brain, which really have a low efficacy. There's a high placebo effect. The antidepressants in general only really, in the end, help about 30% of the patients who take them. And there's resistant populations. And so with increasing depression by COVID, no new mechanisms, that psychedelics really appear to be a really exciting development to treat depression. And some of the early clinical data suggests that 70% of patients, 70 to 80% respond to psychedelics, and that a single treatment has antidepressant effects, prevents somebody from relapsing for 3 to 4 years. So it's really a paradigm change, going from taking a drug every day that may be only helping 30% of the people to 1 or 2 treatments helping 70 to 80% of the people over several years. Represents a real paradigm shift in psychiatry.
Outside of kind of like altering mood, are there any other kind of applications of psychedelics? Like, I think you mentioned kind of like its anti-inflammatory effects. Could that kind of be harnessed essentially?
So that's a main discovery from our lab that we're really trying to develop into therapeutics now. We've discovered in several animal models that psychedelics are really potent anti-inflammatories at sub-behavioral levels. And we've looked at models of inflammatory bowel, asthma, cardiovascular disease, ocular disease, and it's really potent suppression of a lot of symptoms and pro-inflammatory biomarkers. So what psychedelics might also represent sort of at sub-perceptual non-psychedelic doses would be potent anti-inflammatories to treat things like inflammatory bowel and asthma, arthritis, diseases like that, apart from psychiatric disease. But then there's a fairly substantial fraction of people with psychiatric disease where inflammation is involved, neuroinflammation for schizophrenia, for psychosis, even in drug abuse, substance use disorder, there's neuroinflammation associated with that. So not only could could the psilocybin be directly affecting neural processes and circuitry to treat things like depression and substance use disorder, but the anti-inflammatory component may also be a key factor in treating these psychiatric diseases.
If we kind of look at it from a genomics perspective, what kind of research it kind of is out there that's showing the effects of psychedelics on kind of gene expression changes?
So there have been a few groups, including mine, that have looked at this, and we initially started looking at the effects of LSD and other psychedelics on gene expression using like the first generation Affymetrix arrays. And surprisingly, in those first studies, we found that what I would consider a whopping dose of LSD in the rats had a really small effect in the brain at that size. We only were able to identify and verify about 7 to 10 genes whose expression was significantly differentially changed that we were able to validate out of, I think, 3 different, the Affymetrix A, B, and one of the other chips that we screened. So that was somewhat surprising. But then what we also found was that almost all of the genes had something to do with synaptic plasticity and facilitating how neurons communicate with one another. We saw no genes that were indicative of damage or stress or toxicity. It was really a small collection of genes that were involved in synaptic plasticity. And some other groups also did genomics, RNA-seq, microarrays in mice, identified similar things. So we followed up that with looking at persistent effects of LSD in a model of psychosis, where we treat the animals every other day with a low dose, and it essentially made them go crazy over 3 months. And we are one of the first groups to apply RNA-seq to behavioral changes induced by LSD. And we found that even in the absence of the drug, that there were over 700 genes that were significantly differentially expressed by this chronic treatment with LSD. So that shows us that for better or worse, that psychedelics can influence gene expression within the brain, but that how it's influencing it to be potentially therapeutic versus pathological really depends on the amount and the dosing.
You mentioned the kind of long-term effects. Are other kind of omics technologies being applied to explore some of the other kind of ways that it's being affected? For example, like the epigenetics and kind of that area.
Yeah, so that's what one of the main projects in my laboratory right now is. We are trying to identify why a single dose of psilocybin has antidepressant effects. And with ketamine, for example, it's antidepressant, but it wears off after about 2 weeks. With psilocybin in humans, it's demonstrated to last upwards of 4 years, if not more, for a substantial number of patients. So why is it so long lasting? We don't really know that. The first thing that would obviously jump to mind is that there's some epigenetic changes that are occurring. And what we have developed is a rat model where a single administration of psilocybin has very long-lasting, persistent antidepressant-like effects. So we've taken the brains of these animals, we've dissected out key brain regions, and we're in the process of preparing the RNA to do RNA-seq, to do ATAC-seq, to look at gene expression and epigenetic changes. And we're also now considering how can we leverage the single-cell RNA sequencing into this pipeline. So we've been looking at that. We've developed protocols called neurocytometry that allows us to go into banked fixed brain tissue and sort out individual cell types based on flow cytometry. So what we'd like to do is see if we can combine that with the single cell techniques to where we can get these entire populations of brain cells from our fixed brains and do profiling of, say, what's in the medial prefrontal cortex. How are we potentially changing the population of maybe microglia between the depressed and the non-depressed and the psilocybin treated. So also potentially combining that with the single cell ATAC-seq to look at epigenetic changes, and maybe we'll get some clues there.
Yeah, I mean, yeah, because that would make sense. Like applying like kind of single cell and spatial technologies would be good in that area, because obviously it's targeting such a kind of important parts of the brain. But what are the kind of challenges of that? Because like obviously there's not a lot of brain samples kind of going around. How are you kind of dealing with those challenges?
I think one is just having a good animal model that I think has good validity, construct and predictive validity. And I think with our rat model that we've developed, it's a stress-based model that's effective in females, which most of the animal models of depression aren't effective in females. So, which depression in the human population is more prevalent in females than males, but most people are studying male animals. So we've got a female depression model that's based on stress, where a single dose of psilocybin is really able to normalize specific brain circuitry function and behaviors. And so I think going in with a good animal model is paramount. The other thing is our our process that we developed for neurocytometry, to we're able to get single cells from these brains, as opposed to just getting sort of unlabeled nuclei. We don't know what they're for. So we're able to put in a flow cytometry step where maybe we can select for only neurons or interneurons to do the single cell sequencing to get it a little bit better granularity or detail than just taking the entire brain as it is. So that's one of the things that we hope may give us some clues by sort of going in with the targeted cell population to begin with.
Are there kind of any other areas that your group or any other groups are kind of focusing on?
One of the other areas, in addition to the antidepressant effect of psilocybin, is the immune cell function and how psychedelics are affecting immune cell in our model of asthma. So we're looking at macrophages and T cells, and that's another area that we want to apply genomics to, is looking at how we're affecting the immune cell landscape within these tissues, for example, the lung, and doing single cell PCR and genomics and single cell ATAC, looking at how psychedelic is affecting, say, Th1 cells or Th2 cells in the lung that's asthmatic, that has been treated with psychedelic or not, towards identifying what the therapeutic mechanism is. The mechanisms right now are really unknown, but we do know that we are suppressing cytokine secretion from innate cells, from adaptive cells. We're preventing recruitment of eosinophils and T cells to the lung, but what those mechanisms are and why 5-HT2A receptor activation with psychedelics, but not serotonin, is still a mystery. So I think going in with some of these newer genomic profiling paradigms, we can really assess sort of the landscape of what's happening within these tissues at a detail that we wouldn't normally be able to.
Are there many kind of groups kind of studying this kind of area of research? Because I can imagine like funding is kind of difficult to get just because of like kind of previous like kind of thoughts on the use of psychedelics and that kind of research area.
Most of the work is done by clinicians and psychologists and psychiatrists who are looking at depression and substance use and patient response there. There's very few people who are looking at mechanistic aspects of how this is happening. There are a few groups. There's myself. There's Dr. Gonzalez Mesos, who is at Virginia Commonwealth, who's also doing some genomic studies, and they've done some epigenetic studies looking at 5-HT2A receptor and psychedelics and schizophrenia. So I think actually doing genomics in the field of psychedelics, that I can think of, really only about 2 labs right now that are doing that. I think it's difficult to get the necessary permits to get these drugs to do the research, but that's becoming a little bit easier as more people are coming into the field. I think still a lot of it is clinically focused, not so much on the basic science.
How is your kind of research, say you're in the lab and you're doing your epigenetics and you're doing the single cell and stuff, how is that then kind of being translated, I suppose? Because I mean, you could find something really, really exciting, but then how is it going to impact kind of drug development, I suppose?
So what I think is, if we can find and what the mechanism is, so we do our single cell sequencing, We do our ATAC-seq, we find a collection of genes that are surprisingly differentially regulated with respect to, say, antidepressant effects. And it turns out that that's a key mechanism for the antidepressant effects. Then we combine that with our receptor pharmacology, drug design and development, and try to develop a new class of psychedelics that are activating the receptor that will preferentially only activate the effector pathways necessary for that therapeutic effect. So we could conceivably design drugs that only activate these antidepressant pathways, or we could identify drugs that would only activate the pathways leading to anti-inflammatory effects, which I think are probably different. So we could really tailor new chemical entities and drugs that activate the receptor to really only selectively activate whatever that therapeutic pathway is for that disease indication, which would mean less side effects, less behavior effects for people taking the drug for other indications. Because if you don't need to hallucinate, why should you hallucinate for therapeutic effect? And we can engineer that out, I believe.
Yeah, I mean, is there a way to kind of synthetically engineer novel kind of like, not products, like drugs? Because I saw something like to do with like mushrooms, and they said that they found that a particular gene that was in kind of involved in it, in its effects, and I suppose that you could eventually, I don't know, find whatever gene is there and kind of synthetically engineer. I don't know if I'm making that up.
Yeah.
I don't know, like how could that research kind of progress the field, I suppose, because I feel like that's like a really exciting area.
Well, there's another area, Brian Roth, who I think he's a third laboratory who's working on the molecular biology and receptor pharmacology of psychedelics, not so much the genomics part yet, but he's been given a $26 million grant by the US Army to essentially figure out how to make a non-psychedelic antidepressant. So they're doing a lot of really cool stuff in his lab. But I think it's having a basic understanding of what pathways are being activated for drug development is really sort of the direction that it's going. Like we've developed some new drugs in my laboratory that are as anti-inflammatory as our prototypical drugs, but are less behavioral. So they're not making the animals have the hallucinogenic behaviors in the typical assays. So rather than having, say, psilocybin that everybody takes, okay, that works, but people are going to have to hallucinate. They're going to have to be in a clinic for 3 or 4 hours. But what if you could make a drug that is just going to be targeting specific pathways that receptor could possibly activate out of the many that it can, that will just selectively activate whatever it is that's causing the antidepressant effect or the anti-inflammatory effect. And that could be different between different diseases, like to treat asthma, to treat drug abuse, migraine headaches, might all be a different pathway that this receptor is acting at. Yeah,
I mean, currently psychedelics are often used in kind of a recreational like capacity, but how can we kind of ensure that there is like control over the use of this? And I know often people go on about like micro dosing it and that kind of stuff.
Right. I think what MAPS is trying to do with MDMA and have these treatment therapy centers, I think there's going to have to be, it's really a new paradigm for the FDA and for the DEA to consider. But there have been public presentations by both the DEA and the FDA that they're both amenable to developing psychedelics as therapies. Like the FDA is on public record at several conferences for saying, yeah, if you can show that these drugs are safe and effective, we will work with the DEA into revising scheduling laws and coming up with proper regulatory protocols for the use of these drugs. And the DEA has also said, yes, show they're safe and effective, and we'll work to see how we can make this happen. So I think that as things are progressing, it's really unknown territory, that we're going to have to come up with new treatment strategies, regulatory affairs, on how to handle these. And I don't think that you're going to see where any psychiatrist or physician will just be able to prescribe these drugs. That I think there are safety and regulatory issues that need to be taken into account. For example, there are people out there that should not be taking psychedelics, primarily those that have a history of psychosis in their family, that there's a chance that it could make that worse. And so there have to be rigorous, I think, screening protocols in place to make sure that only people who are really at low risk for, say, making a psychosis worse are getting these therapies. So what the prescribing laws and everything are, I think, remains to be seen. And it's kind of going to be developed along as we go.
Yeah. What do you kind of think the future of this field kind of looks like, and what are the kind of challenges that remain?
Oh, the future, I think it's promising. I know when I got into the field, there were maybe half a dozen researchers, and my father was one of them, and he told me it was going to be the kiss of death if I went into this for my career. And I ignored his advice. It's going from where it was, half a dozen people to now so many. There's hundreds of people and companies that are now in the space. It's kind of like the Wild West. That I think there's going to be a lot of good that comes out of it, a lot of crazy stuff. But I really hope that that the therapeutic potential of these drugs can be really vigorously and scientifically vetted, and to where they're accepted as therapies. And I think this is a real possibility in the next, say, 5 to 10 years, that we'll see therapy centers with MDMA, with different psychedelics. We'll have the proper protocols out there, treatment centers, and really be able to help people. And I think what started for me as a curiosity is, how are these drugs working, to now having a real legitimate chance of helping alleviate suffering is I think a really positive thing.
Thank you so much for joining me today, Charles. It's been really interesting, and I think this kind of area of research definitely doesn't receive kind of a lot of attention, and I've definitely learned some stuff today, and I hope others have too as well. So thank you so much.
Thank you.
Hello everyone, if you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website, frontlinegenomics.com. I hope you enjoy.